All right. Hi, everyone. I hope that you are all doing well. So right before I came over here, I sent you all an email with a link for a doodle poll for the review session. Please reply to that before Wednesday's class. As I mentioned in the email, the times start on Friday, only because I have a limit to how many times I'm allowed to have in Doodle. They don't let me put infinite times in. So if you really want something before that, send me an email to vote for it. So today I'm going to talk about transplantation. Please remember that you have the clinical immunology assignment due today by 5, if you haven't already submitted it. And in addition, there is the vaccines assignment due on Friday. So just be aware of all that stuff. The final exam is a week from today. And yeah, I will go put my head down into grading mode after I'm done with this. Yes, Emily.
So genuinely for the assignment today, I know the examples have like one sentence.
Yeah,
That's
totally
all
fine.
you need.
Totally fine. Yep.
So it would like be like one page of just like blah 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 blah.
Completely fine. Yes, that is completely fine. All right, so today I'm going to talk about transplantation and transplantation immunology. And I'm going to hope that this works. Okay, so there are a lot of things that one could talk about when one is thinking about transplants and organ transplants. So one thing that I will say only a little bit about is blood transfusion. And you can see some examples of some red blood cells up in the upper right, where we are seeing a transplantation of red blood cells, as we see in blood transfusion. We also will sometimes see situations where there are bone marrow transplants. Both of these things are sometimes are sort of special cases a little bit. Much of the time when we are talking about transplantation, we're talking about organs that are shown on the left side of this, which are also sometimes referred to as the solid organs or solid organ transplantation. You can see that the most frequently transplanted solid organ is a kidney, mostly because we have 2 of them, so you can get them from living donors. You can also see that there are sometimes transplants of heart, lung, intestine, pancreas. This doesn't show cornea, but cornea could be on here as well. And so we're specifically thinking about issues related to the immunology of these sorts of transplant situations today. As you will see, a lot of what we're going to talk about is specific to the left side, to sort of solid organ transplantation, which has its own specific set of sort of general issues. There are unique things that come into play when we're talking about either blood transfusion or bone marrow transplantation, which we'll get to a little bit later. But if I am not specifying, I mean solid organ transplantation. And by and large, when I give you an example, I'm going to be saying the kidney that we're transplanting, because kidneys are just so much more common than the other types of solid organs that are transplanted. So let's sort of think for just a second about the details of what might happen with kidney transplantation. In general, when we have a transplant from any type of donor, we need to have an organ that is in very good condition. So it either needs to be coming from a living donor or needs to be coming from a donor sort of immediately upon their death. because we don't want the organ to lose blood flow, to start to degrade and have damage. And so you can see a situation where we might have a person who passes away right away, and thus we're going to want to get organs from them immediately upon their death. You will see that, we call our person who is in need of a kidney in the middle of the night. We fly that kidney, to wherever the person is, and we go straight into surgery, because this person, while they are lacking this organ, is going to be having some pretty severe medical issue that requires them to need a new organ. And so this needs to be done ASAP with this organ sort of as fresh as possible. You can see there's kind of a whole lot of work that's going on here. And sometimes all that can be ruined, because when we transplant in that organ, there can be an immune response where T cells will actually kill the kidney after we put it in following this transplantation. And so understanding what this immune response is critical for helping our transplant patients have successful transplants and successful treatment. And thinking about, knowing how this process works also may allow us to do some types of treatment. And so kind of, I guess what I would say is that you should realize kind of the stakes are, there's stakes on this one that we should be aware of. When we look at traditional solid organ transplantation, we notice that we see hallmarks or features of an adaptive immune response, indicating that some aspect of adaptive immunity is sort of what's going on, what's the problem in this type of response. In this slide, you're seeing experiments where investigators are taking skin from one animal and transplanting it onto another animal. So they're referring to it as the skin graft. Whenever we talk about transplantation immunology, we sometimes will use the term graft. Graft just means the thing that is getting transplanted. And so if I was talking about a kidney transplant, I could also talk about that as the kidney graft, the kidney that was transplanted. So here you see a skin graft. And you can see the situation where if we have a mouse of strain B, which I know both because it's labeled and because it's sort of a mouse that's shown here in white, and it gets a skin graft from a different mouse strain, a genetically different mouse strain, which I know it's labeled as strain A and it's sort of shown in gray. We will actually see that grafted skin get killed in about 2 weeks, and basically that new skin will start to die, and we will have this rejection process happening. And so that 2 weeks starts to make us think a little bit about, hey, this sounds like adaptive immune responses. But what we can also find out is if we take that same mouse who has already rejected, and this is a strain B mouse, that mouse has already rejected skin of strain A, and we give that same mouse a second graft of strain A skin, that mouse is going to reject that graft even faster. And now we're going to see rejection in something like 6 days. And so this is one way that we can say, oh, this must be something going on with an adaptive immune response, because we're seeing this improvement and this memory response with second exposure to foreign skin. People have also done a lot of experiments like what is shown on the bottom here. So if we look at our strain B mouse who has rejected some strain A skin, we could do an experiment very similar to experiments that you saw at the beginning of the semester done by Von Behring and Kitasato, where we could take either antibodies from this mouse or T cells from this mouse and give those antibodies or T cells to another naive mouse. And so the idea here is that we're looking to see, are the T cells or are the antibodies the things that are really important in this rejection process? So this mouse has never actually gotten a skin graft before. It's just gotten either antibodies or T cells from this experienced mouse. And when we do that and we transfer T cells, we find that T cells are the ones that transfer this fast rejection. And so in fact, that it looks like this response is really a T cell mediated response. And in fact, for the most part, transplant rejection is a T cell mediated response. We now know that there are really 3 different types of transplant rejection that can happen. So there are 3 different mechanisms that occur with transplant rejection. They are classified based on how quickly after the transplant the rejection happens. And so the quickest one is called hyperacute rejection. Then the next one is called acute rejection, and the slowest one is called chronic rejection. And what is interesting to notice is that the mechanisms of these 3 types of rejection are actually the same immune mechanisms that we have already seen for different hypersensitivity and autoimmune responses. So you can see that hyperacute rejection is going to have a lot of similarities to a type 2 hypersensitivity response. Acute rejection is going to look like a type 4 hypersensitivity response. Chronic is going to look like a type 3. Now you might say, hey, Dr. Barker, I'm a little confused. You just were kind of telling us T cells were important. And 2 of these, type 2 and type 3, are both antibody-mediated responses. But remember, they're both IgG-related responses. IgG requires a B cell to have class switched and to have gotten some T cell help. So there is sort of this important role of T cells. Though nobody ever thinks about the T cells in type 2 hypersensitivity, and I'm not going to say a ton about the T cells in type 2. You can sort of imagine that's part of what's going on with the T cells, sorry, with hyperacute. So as I said, hyperacute rejection is one that is usually one where we think about having antibodies really be the key. And a hyperacute rejection looks a lot like a type 2 hypersensitivity response. So we know we've got antibodies here. And in general, these are IgG antibodies. And with either a hyperacute rejection response or a type 2 hypersensitivity response, we have antigens that are on the surface of cells. And so basically what's happening is that antibodies are, these IgG antibodies are binding to their antigen on the surface of cells, and then the cells are getting killed. The key thing with hyperacute rejection, of course, is that it's happening fast. We're talking within a couple weeks. And the key reason why hyperacute rejection is happening is because of a person having pre-existing antibodies to the organ that is transplanted. So this would be a situation where, say, if Julie was getting a kidney from Aurora and Julie somehow already today, before this transplant, has antibodies against Aurora's kidney, these pre-existing antibodies. And if you think about it, you're kind of like, why in the heck would I, and these are IgG, they're usually class switch, they're usually pretty high affinity, pretty good antibodies. And you might think about it, why would I ever have pre-existing antibodies to someone else's kidney? I have likely not been immunized with someone else's kidney. I have not been infected with someone else's kidney. Like, why the heck do I have these pre-existing antibodies that have definitely come from some general type of process that, you know, had to involve some previous immune response to that person's antigen? And also, I will just note here, you can see, oh look, There are the antibodies that are binding to like the capillary wall of the kidney and destroying the capillary wall of the kidney. And then the kidney doesn't get blood because the capillaries are not working. So the question sort of comes into how in the world is it possible to ever have pre-existing antibodies against another person's organs? You can also sort of see this same process here. This person had pre-existing antibodies, clearly not to scale, that could potentially bind to the kidney. They immediately bound in the vessel of the kidney, led to occlusion of the vessel, and that kidney was no longer able to persist. The kidney fails, and the patient now is in kidney failure, and unless they can get another kidney, which was already hard enough to get them the first one, they're probably going to be pretty sick, potentially die. What in the world? Excuse me for a moment. Those are so, wow. I didn't put the slides in here that I wanted to. So, let me just tell you in words why you have antibodies to another person's antigens and what exactly is going on here. So the real issue here is that we only tend to see hyperacute rejection with one particular type of antigen, which is an antigen that you do have against other people's tissues. And those are the blood group antigens. So if you recall, I told you last time that there are some people with type O blood who basically don't have particular terminal sugars on their red blood cells. Some people have type A blood, and so they have a particular type of terminal sugar on their red blood cells. We have some people who have type B blood. They have a particular type of terminal sugar on their red blood cells. And we have people with type AB blood who have both terminal sugars on their red blood cells. Okay, so these are all of the blood cells of people based on whichever blood group antigen they have. And remember that when we talked about the different blood group antigens last time, I told you that they were carbohydrates, different types of carbohydrate antigens. So one could make antibodies against these antigens. And in fact, because VDJ recombination gives every combo, developing in your bone marrow, everyone will have B cells that make antibodies against the A antigen and B cells that make antibodies against the B antigen. So in the bone marrow, everybody will have B cells with these specificities generated. So in the bone marrow, everyone makes all these B cells with all these specificities. But then what happens in the bone marrow of this person with the A antigen on their red blood cells? And also remember, red blood cells develop in the bone marrow, so there's plenty of them in the bone marrow. What happens here to this B cell as it develops? Yep, David.
The cell that makes that antigen basically gets cold.
Yeah, so we're going to delete this cell. So it's not actually going to leave the bone marrow. You're never going to make those antibodies. What's going to happen in this person? This B cell's deleted during development because it's self-reactive. What happens in this person? They're both deleted because it's self-antigen. So you can see that basically there are sort of differences in what kind of antibodies you have against these blood group antigens based on your blood type and which things are self-antigens. So the thing that sometimes people sort of get confused about here, you can find this in like many intro bio textbooks, like some version of this type of thing. And so you could imagine, for example, you could say, oh, well, a person who is type O can donate blood to anybody because their red blood cells don't have any antigens on them. So those red blood cells are not going to have a problem here in the face of anti-B antibodies. They're not going to have a problem here in the face of anti-A. You could say, oh, AB blood cannot go into a B person because there's antibodies against it. You could say, oh, AB blood can't go into this person with A because there's antibodies against it. AB can't go into this person with O because of the antibodies against it. This is where we get this idea of the universal recipients and universal donors and all that kind of stuff. The one thing that sometimes people, like I said, get confused about, the one part that doesn't show up in intro bio textbooks that I will admit confused the heck out of me as a beginning immunology student, was the idea that, like, yes, you can do blood typing in the lab in like 5 minutes. It's super easy to do. So I blood typed myself one day in the lab as a demo for students. I found out I'm type B blood. I have type B blood. I can understand pretty easily why I don't have antibodies against the B antigen. The thing that sort of confused student me was, why do I really have antibodies against the A antigen? Like, yeah, I made that B cell, but I also made a B cell that responds to Ebola, and I never had Ebola, and I don't have a lot of Ebola antibodies around. That's like the whole point of like why you need a vaccine and stuff. So why do I have a whole bunch of pre-existing antibodies against the A antigen? Somehow, my B cell that makes this antibody has been triggered in the past, has class switched, is making huge amounts of antibodies that will keep me from ever receiving blood from someone of type A or type AB. Like, I can't receive blood from them, and it's because I made these anti-A antibodies already. It's these pre-existing antibodies. The reason is because these antigens are all carbohydrates that are also commonly found on bacteria in your GI tract. And so if any of these B cells make it out of the bone marrow, those B cells will see an antigen with this carbohydrate, and they will make a lot of antibodies. And so you, in fact, naturally, if you are a type O person, are going to make a ton of anti-A and anti-B antibodies by responding to the bacteria in your GI tract, and so on. And so you have these pre-existing antibodies to these different antigens that are on other people's cells already because of this process. And these pre-existing antibodies would be the key drivers of hyperacute rejection. Hyperacute rejection does not happen very often in modern times. How might you prevent, and how in fact do we prevent, hyperacute rejection? What do you think, Roxy? Blood typing. We do that 5-minute blood type thing that I told you about, quick and easy, and we're able to make sure that we don't have hyperacute rejection based on what we know about sort of these blood group antigens. In general, we blood type, we avoid all hyperacute rejection because we just check beforehand and limit the what we're doing in transplantation. And to be honest with you, at this point, at least in sort of, I was going to say modern Western hospitals, but I'm not sure how many non-modern, non-Western hospitals are doing transplantation. At this point, if hyperacute rejection happens, there's generally a ginormous lawsuit. And like hyperacute rejection should not happen because it's pretty straightforward to deal with. However, if the graft does not get rejected by hyperacute rejection, in the first week or two, then the next thing that graft is susceptible to is acute rejection. So we're going in order of timing. And acute rejection, as you can see here, is very similar to a type 4 hypersensitivity response. It's going to largely involve a lot of T cell responses. And when most people think about transplant rejection and sort of what's going on with the immunology of transplants, they're thinking about acute rejection. It's the sort of predominant part of transplant rejection. And so here you can sort of see this situation of acute rejection happening. And what you will notice, as you can even see from my title here, is that acute rejection is super dependent on T cells. And so what we'll have happen is we'll have our transplant, like our kidney graft, that will be attached to the blood vessel of the rest of the body. And at some point, we're going to have some T cells come into that kidney graft and start to destroy that kidney graft so that it is no longer going to persist in the patient. And in fact, in these images that you see at the bottom, you can actually see T cells coming in and invading a kidney graft before rejecting it. And so the idea is that these T cells are rejecting this graft because they are recognizing it as foreign. Just like with the type 4 hypersensitivity response, there are a couple different phases that we see with acute rejection, known as the sensitization phase and the effector phase. With the type 4 hypersensitivity response, a lot of times we talk about those two things as happening at different times, like the first time you have poison ivy versus the second time you have poison ivy. And that's because you don't have like permanent poison ivy. You're not like, I don't know, you're not wearing like poison ivy clothes every day so that you're always seeing that antigen. In the case of transplantation, that kidney's with you 24/7 every day. So you don't really have, there isn't the same amount of timing separation between these two phases, but the same two sets of events do have to happen. So first, we do need to have some antigen from the kidney. Yes, I am being very purposely vague right now about the antigen, and we will get more into the antigen going forward. There is some antigen from the kidney that is going to make its way to a lymph node. Once in that lymph node, T cells that respond to that antigen are going to get activated. They're going to become effector T cells, and so we're going to see this T cell activation in the lymph node during the sensitization phase. Then those T cells, once they've been activated, will leave the lymph node and will actually head back to the kidney and start to actually kill the kidney or kill the graft. So this is the rejection phase or the effector phase. It's, again, it's going to be a bit slower we're probably talking about months in certain situations, or maybe a month to a few months, as opposed to the other one, which was like a week. But it's not quite as separated as it would be if this was an allergic reaction. So this is where I get to, this is always a little bit of a tricky part of the semester, but that's okay. So now we got to think about what's the antigen that's actually being recognized here. And early immunologists actually figured out a whole bunch of parts of this. They in fact were doing experiments where they were grafting skin on different mice of different genetic backgrounds to try to understand the genes that were really important in transplant rejection. And in fact, they found that the gene that was most important, I said most important for a reason because there are other ones too, but we'll get to those. But right now we're going to say most important, was the MHC gene. In fact, the M in MHC is major because it's the most important, but not the only important one here. And so they found that if you had a mouse and you gave it skin from a genetically identical mouse, then in fact you would not have that skin be rejected. So in fact, if you do any transplant between one person and someone who is genetically identical to them, you don't see rejection. Sometimes we refer to those grafts with a prefix of syn or auto. So sometimes we'll talk about a syn graft or a syngeneic graft or an autogeneic graft. The idea with either of those is you are going between two individuals who are genetically identical. Pretty easy to do in mice, not quite as easy to do in people. And so you can see this syngeneic graft, not rejected, so MHC matching. If, however, MHC does not match, then in fact that transplant gets rejected. So you can see that we're getting rejection of this transplant. We're seeing that the transplant doesn't stay. And in this case, again, you can see we have this prefix. Now instead of syn or auto, we're using the prefix allo. So this is an allogeneic graft. Syngeneic is genetically identical. Allo means that we're talking about genetically different. Member of the same species. So we're going from a mouse to another mouse, but they're not genetically identical here. And in general, if you are getting a transplant from another person, it's going to be an allogeneic transplant because it's from another person, but they are not genetically identical to you. And you can see that if the MHC doesn't match, that graft is rejected. And in fact, the main antigen here is MHC. That's how it got its name, the major histocompatibility complex. It was the main gene responsible for whether tissues could be transplanted, was really how it was discovered. And they could also do some experiments like this, where they could have a mouse of one strain. They could see that, and so we could also have a recipient that was a child of two inbred strains. So we had a strain B mouse planting to its child of strain A and B, and there was no rejection. So we can see that, as long as there was some matching, it worked. But we could also see that if the child tried to transplant back to the parent, we saw that it didn't work. If there was any foreign MHC present, then we weren't going to have transplantation working. So we saw this rejection phenomenon. Please note that this data that you see here on transplantation of, whether you can get a transplant from your parent, whether you can get a transplant from your child, etc., is true in inbred mouse species. When we go to outbred species like humans, it gets a bit more complicated. And so please do not take this to just go call your mom and be like, "Ma, give me a kidney." You totally can, because it doesn't work that easily in outbred species like humans. Yes, Jazz?
I was going to ask, because from this, theoretically, your best match would be like somebody who's your sibling because of recombination from both your parents. And your parents really couldn't donate to you unless they themselves had identical MHCs, which is unlikely since you're most attracted to somebody who has an opposite MHC. So like,
We're getting there.
okay.
And so one of the answers to Jazz's question is that what we usually try to do with a transplant is we try to match as many MHC molecules as possible. And so this is looking at matching of MHC class 1 between a donor, a transplant donor and a transplant recipient. And so you can see that if we have 0 mismatches, then things work out the best. The transplant has a half-life of usually about 13.4 years. We've got this 10-year estimate. Whereas if we have 6 MHC class 1 mismatches, we got a lot bigger problems. So part of what we do, Jazz, is we try to find someone with the highest number. We try to minimize the mismatches as much as possible. This is part of why, back to that figure at the very beginning, it's really hard to find a match for a transplant. This is why people wait years on transplant lists looking for donors, and why people go on TV and it's a whole big thing, because you need to find someone with a very similar MHC to yours and with a match ID with as many MHCs as you possibly can have. When I decided after that doctor told me that he thought he knew my MHC type by looking at me, I of course then went into Googling how if I could actually find out my MHC type myself. And it turns out the way that most people find out of their MHC type, if they find it out, is because they sign up to maybe be a kidney donor or something like that, and they get typed for those kinds of reasons. And so sort of making sure we have matching MHC is sort of a really key piece here. But this is the slide. So I showed you this slide earlier in the semester. And I specifically was telling you about this slide with understanding MHC and MHC restriction. And I talked about it with the Zinkernagel and Doherty experiment. Where, if you recall, Zinkernagel and Doherty found that T cells could recognize antigen-presenting cells that had the correct MHC and the correct peptide, and that if a T cell saw the wrong MHC, it didn't work. If it saw the wrong peptide, it didn't work. And that was true. That's absolutely what they saw, what they won the Nobel Prize for. That's how MHC restriction works. Zinkernagel and Doherty, we now know, were super lucky in that experiment. In the types of mice they happened to pick genetically to make this work out perfectly. Because it turns out there's actually a little bit more of a complication in all of this than we talked about before. Because if you look at this, you might be a little bit confused. And I had somebody come ask me questions about this earlier in the semester. Because I have just told you that acute rejection happens because of T cells and MHC being mismatched. And now we're saying if the wrong T cells see the wrong MHC, they don't do anything. But then I just told you like a few minutes ago, if T cells see the wrong MHC, they kill the kidney. So like, what's up? It's a little bit tricky here. And this sort of does get to the fact that when we talk about this first sort of pass through MHC restriction, we are oversimplifying a little bit. And Zinkernagel and Doherty, like I said, lucked out in terms of the mice they picked, giving them this perfect, simple answer. In reality, so if you look at this image, you can see the MHC and the peptide. And they're like nicely color-coded and labeled, and it's really easy to know what's MHC and what's peptide, right? In this image. This is actually what the T cell sees. And it's not color-coded for the T cell. There's just sort of this surface of atoms that the T cell is looking at. On the left, you're seeing an MHC class 1 molecule with a peptide. On the right, you're seeing class 2 with a peptide. And really, it's just this 3D surface of atoms that the T cell is seeing. And so sometimes we'll have a situation like the one that's here on the left, where we've got this self-MHC, and we've got a peptide. The two of them come together to make a three-dimensional shape that the T cell recognizes. Easy. Sometimes we'll have a situation like this situation here, where the MHC is so dramatically different from the other MHC that the T cell cannot get confused. That the T cell cannot even slightly start to react with that configuration of atoms. That the MHC type is just so different that there's no possibility of reaction. This is what happened with Zinkernagel and Doherty. They didn't know this when they did the experiment. It's just they happened to pick two mouse strains whose MHC types were so structurally different that the T cell had absolutely no way of recognizing. More often, however, we might see some MHCs that are not quite so different from the self-MHC. So here you can see the self-MHC is this green one. It's got two little ball things on the side and nothing in the middle. Well, here's this purple one. It's got two little ball things on the side and some stuff in the middle. It's kind of similar structurally. It's not like crazy different. This one has, again, the two little ball things on the side and like one little thing in the middle. And so sometimes if you actually add up the whole surface of the MHC plus the peptide, it can actually look like the same 3D surface of some other T cell plus peptide. And so here, notice that this T cell is responding to parts of the green MHC and parts of this brown peptide. So it's these two brown squares and these two little green balls. Well, if an MHC molecule also had those two little circles and had two little squares, just in terms of its structure, it would fool that T cell. And that T cell would actually get turned on by the MHC no matter what peptide was in there. So sometimes MHC can actually sort of cross-react, and you'll get an MHC type that can actually turn on the T cell. Basically, that foreign MHC looks like self-MHC plus peptide. You can also see a situation where that foreign MHC could look like self-MHC plus peptide when it's combined with the right peptide. And so here you can see, the foreign MHC is giving some amino acids, the peptide is giving some amino acids as well. And so sometimes foreign MHC will have sort of an amino acid structure, a three-dimensional structure that will look like self-MHC plus peptide, just because of how the atoms are sort of there in the structure. And so sometimes your T cell will be able to react with some foreign MHC and think that it is a foreign peptide being presented. And so that's really how allo recognition works. And so the idea, what you can think of here is you can think of this idea where this foreign MHC is somehow being recognized as a foreign-looking thing by the T cell. And it might be because structurally it fools the T cell into thinking it's self-MHC with peptide on it. That's one way that this can happen. This method of allorecognition that's shown in this slide is officially called direct allorecognition, which you can see on the left. So here we've got our antigen presenting cell from the kidney. So imagine this is just a kidney cell from the kidney. And in the process of getting transplanted, some of the kidney cells die. There also are probably some like dendritic cells from the recipient or from the donor that are still in the kidney. You didn't like flush every last one out. They might make their way into a lymph node and interact with some T cell in the lymph node that unfortunately happens to be able to react with that MHC and think it looks like self-MHC plus a foreign peptide. And so that's direct allorecognition. We can also see one other way that a T cell can get activated here. And in some ways, this one is like a little bit conceptually easier. It either like is way conceptually easier for you or is baffling. It doesn't seem to have an in between. And this one is called indirect allorecognition. You will also see this indirect allorecognition on a subsequent slide, unless I messed that up too, but I should have that slide. And it's color-coded on the next slide, so sometimes it helps people a little bit more. But the basic idea here is that we can also imagine a situation where, we have this kidney that goes into the recipient. So the kidney comes from the donor, goes into the recipient. There's some, dead kidney cells, right? They slough off, they go into circulation, and they get degraded. When they get degraded, they might get phagocytosed by a cell from the recipient. And when that happens, the recipient's cell, the recipient's antigen presenting cell, is going to degrade up all of the proteins from that kidney cell and might actually present the MHC molecule on its own MHC. Basically be like, look at this foreign thing I found. This is a protein that we did not see growing up. This is a new protein we never saw before that we are showing on our MHC molecule, because MHC is a protein. And so it can get, in fact, processed and presented by the recipient's antigen presenting cell as well. And so you can see the difference that's shown here is whether we're looking at the donor's antigen presenting cell or the recipient's antigen presenting cell being really key. Okay, this is the other version of this. So here, the donor, the kidney is yellow, okay? The recipient is orange. So the person who gets the kidney is orange, the person who gave the kidney is yellow. We could have the yellow MHC just present stuff that happens to turn on the T cells because biochemistry. Or we can have a dead kidney cell, which you can see here, that has yellow MHCs on it because it's from our yellow donor, get phagocytosed, get degraded into yellow MHC peptides, and get put on the orange MHC to present to T cells and be shown as, look at this foreign peptide that I have found. And so this would be indirect allorecognition, this would be direct allorecognition. Are we directly recognizing that foreign MHC without it being degraded and processed, or are we recognizing it having been degraded and processed on our own self-MHC? And so those are really those are particularly key aspects of allorecognition and acute rejection. But if you look at this image, you might come up with another question. You might say, okay, I understand why with 6 mismatches, the kidney doesn't stay very long. I understand why we have MHC mismatch. We're going to have either direct or indirect allorecognition. That kidney is going to get destroyed by immune cells. Got it. And I get that, we want to have fewer mismatches. But what I don't get is if we look at this 0 mismatch situation, it's not as if 100% of the kidneys survive and that we've completely gotten rid of acute rejection. Even if we have a completely perfect MHC match. So we have to think about, well, why are transplants rejected even with 0 MHC mismatches? So I'm going to give you a sort of scenario. Yes, I know I'm being a little silly with my scenario, but I think it usually helps people figure it out. So let's imagine, remember Julie needed a transplant earlier. We do MHC typing of everyone in this room, and we find, amazingly, with this probability, with this number of people in probability, somehow, amazingly, Julie and David have an exact MHC match. Every single MHC between Julie and David is the same. Amazing. I'm going to take a kidney from David, give it to Julie. Can you imagine any other situations where there might be some antigen that Julie might recognize from David? Yeah.
The blood type.
Okay, so I did blood typing. I'm good. I don't have to worry about blood typing. No one's going to sue me for my hyperacute rejection. So it's not hyperacute, not blood type. Why else? Yeah.
Like gender-specific antigens?
So notice I picked a male transplanting into a female, because that's a sort of really obvious one, but there are a bunch of them. David has a Y chromosome, which encodes some proteins. Julie does not have a Y chromosome. I know I'm making a lot of assumptions right now that we're just going to go with, but it's okay. In any case, every single one of those proteins on the Y chromosome is a foreign antigen to Julie. And so any one of those antigens could actually lead to rejection in this case. In fact, there are probably, even if Julie and David were entirely identical MHC-wise, there are other genes that are probably going to differ between the two of them. Unless they are 100% identical twins, there are some genes that vary between the two of them. And there are some proteins that have different sequence. Maybe it's that some actin protein looks a little different from some polymorphism. Like, it could be literally anything. It could be the thing involved with your eye color. It could be the thing, like, you think of a protein, it could be different between two individuals. And so all of those different proteins could also be antigens in this case. These are all referred to as minor histocompatibility antigens, because no one antigen is sort of responsible across the board in everybody's transplant situation. But unless you are getting a completely autogeneic transplant, there's going to be and even if we do a perfect MHC match, we're probably not getting you an organ from someone who is completely genetically identical to you in all ways. And so in the donor, you can see that there's some protein that has, it's a flower with a red thing on it. And in the recipient, it might just be a flower with a blue thing on it. There's just anything protein that differs from person to person. Could be presented on MHC and would be a thing that you haven't done T-cell selection against, and could be a foreign antigen. And so we will see some rejection due to these minor histocompatibility antigens. Usually This rejection is slower because you don't have quite as strong of a response as you do with MHC, which is like all over the place. But we basically always see acute rejection unless you are getting a transplant from yourself. Yep, Chrissy.
So when you are like looking for a donor, do they try and match like genders?
That makes sense to me. When I think about some of the earliest transplants, I think of a bunch of them that are very much not matched and things like that with gender. I'm not sure. I'm not sure if they understood this well enough when they started doing transplants, and then they came up with a other way of dealing with it, and so they dealt with it that way. But yes, that would make sense and would help. It's so hard to find a donor that you probably don't have that many choices. So we've got these minor histocompatibility antigens. You can also see this process here, but yes, Emily.
My question's like about B7. Obviously, you don't have PRRs that are detecting like pathogens. Maybe you do. Like, if there shouldn't really be any pathogens. So like, is it just kind of like during surgery, your body's so like inflamed that?
So, a couple things could be going on. That's a great question. One possibility, so basically, Emily wants to know why those antigen-presenting cells have B7 on them, where the sort of inflammatory or PRR signal came from. One answer could be, there is really no part of your body that's sterile, and so that kidney could have microbes in it. There could be some infection there, even if it's a non-pathogenic infection. But the other one, as you say, is that I think that there's probably a lot of damage. There's probably a lot of inflammation. I mean, the fact that you have to cut someone open and cut out the kidney and some parts of the kidney, you're going to have some dying cells and things, are probably giving you some of those DAMPs, the damage-associated molecular patterns, from sort of the trauma of the process, but that's not particularly well worked out. But great question. So here you can see if we're getting a graft to a syngeneic recipient, then we have 100% of the graft surviving. Everything looks real good. If we have a MHC mismatch, we see a pretty rapid rejection. If we have a minor histocompatibility mismatch, we'll see rejection, but it will be a slower rejection. And of course, this is something we take advantage of in certain situations where, say, we're going to do certain procedures on people. Sometimes we will actually take a sample from them before. This is particularly important with bone marrow types of things. They might take some bone marrow before they do a treatment on you so they can give you back your own bone marrow. You can also imagine situations where they give you, when you have burns, sometimes they will take skin from another part of your body and give you your own skin to try to reduce these issues as much as possible. So then we have this fourth type, or this third type of rejection that also can happen. This rejection happens after acute rejection. So basically, sort of if the graft sort of survives acute rejection, then we can see this final type of rejection, which is known as chronic rejection. Compared to the other two types of rejection, we know relatively less about chronic rejection because it took us a while to get to the point where we could ever have grafts survive acute rejection, to even have chronic rejection really start to become a problem. But it looks like what's happening with chronic rejection is that after transplantation, you start to develop an antibody response to some antigen in the transplanted tissue. And again, you could probably imagine this being any different antigen between two different people. You get an antibody response, it's an IgG response, so you had to have a class switch, you had to have, this takes a while to develop. And eventually those antibodies will actually form immune complexes in the vessels of the kidney or whatever tissue you've transplanted in, and will actually start to destroy the vessels. So it's an immune complex formation, leading to inflammation and destruction of the vessel. This is happening, years post transplantation, but like I said, there's a lot less known about this one because it doesn't happen, or because it's not in a sort of studyable way easily. One thing that we do know is pretty important is probably what has to happen is maybe some of the donor cells have to die, have to get presented to T cells. Those T cells then have to turn on other B cells that turn on other antibodies. And so there's sort of a multi-step process here that's getting you to the point of these antibodies. But note that these antibodies were not pre-existing. You did not have them before the transplant. These were antibodies that you developed much later on after the transplant, while hyperacute rejection had those antibodies that were pre-existing, that you had before the transplant. So there's sort of a whole other problem that comes into play if we start to do transplantation not of a solid organ like a kidney, but we instead do transplantation of bone marrow. And so bone marrow transplantation has its own unique types of issues. And so before I tell you what the issues are with bone marrow transplantation, I just wanted to give you a quick little bit of info about bone marrow transplantation, because you might say, well, just don't do it. Okay, so bone marrow transplantation is really complicated, whatever. It's actually super important in a lot of different situations. So one of them is that many of the diseases that we have thought about this semester, that even might, in some cases, be involved in a homework that's due today, might involve different types of immune cells or immune stem cells that have a problem. If, for example, we have a person who doesn't have RAG1 or RAG2, they're not going to make B cells and T cells in their body. And so the easiest thing we can do is just give them somebody else's bone marrow that has RAG1 and RAG2, and that will grow them a new immune system. And so, in fact, a ton of genetic diseases where you have some kind of genetic issue of bone marrow stem cells are easily treatable by bone marrow transplantation. Many sort of key sort of genetic immune diseases are all bone marrow transplantation types of diseases. The other big thing to think about is that in many types of cancers, which you see on the left, we treat patients with radiation or chemotherapy. And I'm going to talk about cancer immunology on Wednesday, so I'll say a little more about radiation and chemo a little bit there. But the key thing to realize with radiation and chemotherapy in treating cancers is that both of those things are causing DNA damage so that rapidly dividing cells die. Basically, the idea is we're trying to take a cell and and sort of hit it so that all the rapidly dividing cells in your body die. And all the ones that are slow dividers don't die because they weren't trying to divide right then. And that, in theory, that kills the tumor cell because it's a rapidly dividing cell. The bad part is that it also kills other cells in your body that are rapidly dividing cells, like cells in the GI tract lining, which is why you have a lot of nausea, cells in the hair follicle, which is why you lose your hair, and pretty much all of your immune system. So all your T cells, all your B cells, all your everything. So if you get chemo or a bone marrow transplant, you can basically imagine that you're erasing your immune system. I have an image somewhere, I don't think it's here, but I could be wrong, of a person who gets radiation, who first they show him like filled and then they show him empty, of like trying to show you the immune response. And so with all of those treatments, we end up then having to treat our patients with a bone marrow transplant to basically regrow their immune system for them. And so all of these different types of cancers all generally result in someone having to have a bone marrow transplant. Oh yeah, here's the empty guy. And so the basic idea is that we're going to take bone marrow from one person. We're going to have this other person who has an immune system but maybe has something wrong. We're going to give you radiation and chemo. We're going to just remove your immune system. See, you're empty now. We're going to get some bone marrow from somebody else. We're going to transplant it in. It's going to develop in your bone. You're going to basically get a whole new immune system grown in. So there are a couple of things to think about. And the real problem that comes up with bone marrow transplantation is that there is this additional complication called graft versus host disease, also known as GVHD. So GVHD, graft versus host disease, is a complication that's only possible during bone marrow transplantation. So if you think about our kidney transplant, we have the host, the person who got the kidney. That host's immune system is killing the kidney. The host is killing the graft, right? Host versus graft. In bone marrow transplantation, we have this additional problem of graft versus host disease, where basically the transplant, the bone marrow you put in, can try to kill the host. And so what you can see here is we have bone marrow with this mixture of cells that we're going to transplant into our patient. That bone marrow contains some stem cells, as well as other cells, including memory T cells. Because bone marrow has like all sorts of good cytokines. It's a great place to live. It's like where the plasma cells like to live. It's where the memory cells like to live. It's like this great cytokine bank. And so bone marrow is sort of a mixture of cell types. And when you do a bone marrow transplant, you may transplant in some memory T cells in that bone marrow that can go to the host's lymph node. So now these are transplanted T cells, go to the host lymph node and are like, whoa, all of this MHC is foreign. All of this MHC is not okay. And those T cells are then going to go try to basically kill the entire organism because they are going to be activated to destroy all sorts of parts of the organism. And so now it's the immune system of the graft. It's those T cells that were in the graft that are going to try to kill the host. Well, let's see here. I see I have a couple questions. Because I'm guessing what the questions are, I could be totally wrong. I'm going to do this and then come to your questions. And so graft versus host disease can be varied severity. It generally involves a lot of rash. You can see, some of these are basically rash over the entire body surface. You can see a lot of liver destruction. You can see lots of GI tract destruction. Notice that this one is 1,500 milliliters. That's 1.5 liters of diarrhea a day. This is really, really, really severe, and you can see a picture of a kid with graft versus host disease here. And so graft versus host disease is a really severe complication of bone marrow transplantation. I saw Jazz's hand first, and I saw David. Jazz.
In cancer patients, we often have to do conditioning, but in skin patients, would you have to condition too, or no?
Maybe, but there's another way we get around all of this. So the conditioning is not the issue. We're getting there. David, what were you gonna say?
If the bone marrow allo recognition is such an issue, then in cancer patients specifically, during chemo or after chemotherapy, then can we take bone marrow from the patient before chemo extract it, and then when after they undergo chemotherapy and they, right?
Yeah, so in fact, with cancer patients, that is one thing that we often will do, is we will, if we're going to say, okay, you're going to have chemo, we're going to take some bone marrow now, save it, do your chemo, and then we're going to give you your own chemo back, or bone marrow back, so you have an autogeneic graft. But we have even another way of dealing with this, in addition to the things that you guys have mentioned. So if you look at this image, you can see that we've got our bone marrow with this mixed population of cells. So if you were to imagine how you might get around this problem, what might you imagine? You're all way overthinking it. Yeah, Emily.
Like only get the ones that haven't like differentiated into anything?
Yeah, basically take this and purify out the stem cells and just get rid of everything else. So you don't put in memory T cells, just get the stem cells out. And so most of the time now, we don't do full bone marrow transplantation. We instead do hematopoietic stem cell transplantation, or HSC transplantation. So we'll take bone marrow from the patient, we'll get all of the different cells, and we'll just get rid of the ones we don't want, have only the stem cells so that have not yet differentiated, and put just those stem cells into our patient. And so oftentimes we do hematopoietic stem cell transplantation instead of full bone marrow, and that gets you around the graft versus host issue. One sort of nice thing also is originally the only way we knew how to get stem cells was to like go into like the hip bone or something. It was a pretty, like being a bone marrow donor was a pretty involved process. We've actually found a way that you can get them out of the blood now. And so you can get hematopoietic stem cells out of the blood, and so and be a bone marrow donor or be a hematopoietic stem cell donor much more easily. The one other thing that I will mention is you do still have to do some MHC matching, because if you don't do any MHC matching, you're going to get T cells that respond to the thymus, and you're going to have APCs, dendritic cells, macrophages, that also come from that bone marrow that have a different MHC than the thymus. So dendritic cells are going to present on one kind of MHC. The T cells are going to think self is a different kind of MHC. And that means all the pathogens get a free ride because the pathogens are going to be presented on an MHC that T cells can't see. And so it is very important that when you do bone marrow transplantation, you're doing some matching, but the matching is for a completely different reason with bone marrow transplantation. There are a lot of ways that people try to deal with this. One of the biggest is the fact that we now have immunosuppressive drugs like cyclosporin A. That basically is what allows transplantation to happen. Transplantation was like not a thing nearly to the level it is today without cyclosporin A, because it reduces T cell activation and starts to prevent and slow down acute rejection. And so here you can see, in black, we see how long a transplanted organ would survive without cyclosporin. In blue, you see survival with cyclosporin. You see cyclosporin makes a big difference. And you can also see the number of patients with transplants both before and after cyclosporin. Cyclosporin just sort of revolutionized this whole thing. And so we really sort of became able to do transplantation with cyclosporin. But again, as I mentioned last time, we are broadly immunosuppressing patients. We are basically turning down their whole T cells because we don't want T cells to attack the kidney. But as a result, we're making them susceptible to a lot of things. We are immunosuppressing all the T cells in the body with cyclosporin. And so trying to think of ways to do this more specifically is important. There are also a couple of other kind of sort of novel, either policy or other things people are doing. Either one of them, that's just sort of the policy thing. In general, this is a problem. The number of people who want transplants is shown in red. The number of actual donors and organs we have that are available is in yellow. So you can see way more people need organs than we can give organs to. And you gotta make sure they don't fail through rejection. And so we also have been thinking about kind of other ways to deal with this. One of them is a policy thing in terms of how we do organ donation. In the United States, organ donation is opt-in. You have to say, hello, I would like to be an organ donor. You have to check a box. It ends up that it's a pretty low percent. I don't remember the percent in the US who are organ donors. In some other countries, they have opt-out organ donation, where basically you are an organ donor unless you say no. In those countries, it's like 99% of people are donors, because most people just, unless they desperately care, they just don't do anything. So one thing that sort of the policy thing we could just do is change organ donation processes. The other area that a lot of immunologists spend a lot of time thinking about is whether or not there can be transplants from a different species. So now instead of allogeneic or syngeneic, this would be a xenogeneic transplant. So whether we can have xenografts. There have been a few big advances in this in the past few years. It turns out that the animal that everyone really wants to use, while people think about primates, it actually turns out that the miniature swine, they're not miniature. They look like big old pigs. Have organs that are the exact same volume as ours, and so they fit in the space really well. And so people have been doing a lot of genetic engineering and ways to try to figure out ways we could make pig organs that could be transplantable into people. Or also thinking about things like mechanical devices or things like that, though again, there are other issues on those fronts. So I will talk about cancer immunology on Wednesday. Remember the assignment due by 5.